Okay, guys, so we stopped at the carnitine shuttle during the live session on Collaborate, and we will continue from there. These are the three stages of the fatty acid oxidation. We can also call them three stages of the cellular oxidation that we did earlier as well. So stage one is different because the starting material this time is the fatty acid. So stage one is the beta oxidation, but the end product is the same, that is acetyl-CoA. Stage 2 and stage 3 are exactly the same, and it does not depend on what starting material we start with. If we start off with glucose, stage 1 will be different and it was glycolysis, but stage 2 and 3 are the same. Stage 2 is the citric acid cycle, in which we break down the acetyl-CoA molecule, or oxidized acetyl-CoA, and the stage 3 is the ATP production through the electron transport chain or the respiratory chain. So here is the actual pathway, that is a beta oxidation of the fatty acid, and the one shown here is the beta oxidation of a typical fatty acid, that is known as palmitoyl-CoA or the palmitic acid. This one has got 16 carbons in it, and there is a basic pattern of this beta oxidation pathway, and the pattern is that there are four reactions, and they will be repeated over and over again till we break down the full fatty acid. And these four reactions and their sequence you have to memorize. So first is oxidation, second is hydration, in which we add up the water molecule. Third one is again oxidation or loss of electron, and the last one is cleavage, that is breakdown or breaking apart of that fatty acid. This one is also called as thiolysis. So we start off with the 16 carbon palmitic acid, and at the end of these four reactions we have removed two carbons from it in the form of this acetyl-CoA molecule, and the remaining fatty acid, what is left, is has got only 14 carbons in it. So in one round of this beta oxidation, we take away or break two carbon moiety, that is the acetyl-CoA, and the remaining carbons are still intact. So we repeat these four reactions again, and from 14 we get to 12 to 10, 8 in each round, and eventually we break down the whole of the palmitic acid, which had 16 carbons, into 8 acetyl-CoA molecules. Each will have 2 carbons in it. So looking at these reactions in a little bit of detail, first one was oxidation. The actual process that is happening is dehydrogenation. So we are taking the hydrogen atoms out of the palmitoyl-CoA, and along with the hydrogens also come the electrons. That is why it's called oxidation, which is defined as loss of electrons. So we take the electrons out of it and put it in this FAD molecule to make a FADH2. So two hydrogens are taken from palmitoyl-CoA and put it in this FADH, and the result is a trans-enoyl-CoA. The name of the enzyme is dehydrogenase, and this enzyme has got different isoforms available for very long chain fatty acid, for medium chain fatty acids, and for short chain fatty acids. So these different types of fatty acid will have different types of this dehydrogenase, and palmitoyl-CoA was unsaturated fatty acid, and this one enoyl-CoA, that the product we have formed, has got a trans double bond in it. So this double bond has been formed in this reaction. Second step is hydration. Hydration means adding up a water molecule. So water molecule here has been added in the molecule by the enzyme hydratase, and this will result in a hydroxyacyl-CoA as the product, and this will be added to the double bond. So the double bond is again converted into the single bond in the product. Third step is again dehydrogenation or oxidation, very similar to the first step, but the only difference is this time FAD will not take up the hydrogen atoms. It is a NAD positive. So after taking a hydrogen from this hydroxyacyl-CoA, and this NAD becomes NADH. And as I said in the first reaction, when this hydrogen will move, the electrons will also go in this molecule. That is why it's called oxidation reaction. Dehydrogenase again is the enzyme, and NAD is a cofactor. This point you guys need to remember, which is going to accept the hydrogen ion or the electrons. Last step is the transfer of the fatty acid chain, or in simple words, it's called theolysis or breakdown. We are basically going to take a 2-carbon molecule out of this fatty acid and synthesize a acetyl-CoA molecule. So this acetyl-CoA has been broken away from the fatty acid by the enzyme thiolase. Again, absolutely important to remember the name of the enzyme, thiolase. And at the end of this reaction, we will have a 14-carbon fatty acid, which is known as myristoyl-CoA. We started off with the 16-carbon palmitic acid, and after this round of four reactions of beta oxidation, we have reached this myristoyl-CoA, which has 14 carbons, and the 2 other carbons have been incorporated in this molecule acetyl-CoA. So the fatty acid catabolism for energy, how much energy or which products do we get by breaking this palmitic acid. So a thing that I just said in the previous slide, we will start off with 16 carbon palmitic acid. After one round we get to 14, then 12, and so on, and eventually the last 4 carbon molecule will be broken into 2 acetyl-CoAs. In every round we get 1 acetyl-CoA. After a total of 7 cycles, basically this whole process or the beta oxidation reactions will happen 7 times in total. This is important to remember, and we will get 8 molecules of acetyl-CoA in these 7 rounds. Plus in every cycle we make 1 FADH and 1 NADH, so total of 7 FADH and 7 NADH will also be produced. So this is the whole cycle will look like after 7 rounds. 1 palmitoyl-CoA broken to 8 acetyl-CoAs, 7 FADH and 7 NADH. Now in every round we get 1 FADH and 1 NADH molecule. So after 7 cycles we get 7 FADH and 7 NADH. But how do we get these 8 acetyl-CoA molecules? So this can be a little bit confusing. And the answer is during the last round we get 2 acetyl-CoAs instead of 1. So that is why the acetyl-CoA number becomes 8, whereas FADH and NADH remains 7 in the 7 cycles. So the total number of energy that we can get from one molecule of this palmitic acid, how much ATPs basically we can get from one palmitic acid which had 16 carbons in it. We saw in the previous slide that we get 7 FADH and 7 NADH molecules after the full completion. Each FADH molecule is equal to 1.5 ATP, so they will give us total 10.5. One NADH is equal to 2.5, so they will give us 17.5 ATPs. Apart from this FADH and 7 NADH, we saw in the previous slide that we got 8 acetyl-CoA molecules. Now these 8 acetyl-CoAs are going to enter the citric acid cycle, and you'll probably have to open up the citric acid cycle and check what happens to the 8 acetyl-CoA molecules in citric acid cycle. To cut the long story short, in citric acid cycle, when every acetyl-CoA is oxidized, we get 3 NADH and 1 FADH molecule plus 1 ATP. So 8 of these acetyl-CoAs are going to give us total of 24 NADH molecule, which I've just highlighted here, because every round gives us 3, so 8 molecules will give us 24 in total. Plus every round give us 1 FADH, so we will get a total of 8 FADH, and every round of citric acid cycle also gives us 8 ATP molecules, which are right here. So we convert these NADH and FADH also into energy. 8 NADH equals to 20, From the 24 we get around 60. Each FADH is equal to 1.5. It will give us 12. And if we add all of these ATPs up, the total number is what you have to memorize absolutely, which is 108 ATP molecules by one palmitoyl CoA. That is one molecule of palmitic acid. Once it is totally, you can say, oxidized, will give us a total of 108 ATPs in total. Some basic information and location of different types of fatty acids and their beta oxidations, because there are of numerous types basically. Saturated fatty acids, like the one we saw in detail in the previous slides, was the palmitic acid, and that happens in the mitochondria. For unsaturated fatty acids, with that will also happen in mitochondria. Unsaturated fatty acids are the ones that have a double bond or more than one double bond in it. Then there are a certain type of fatty acids that are called odd chain fatty acids, means they have odd number of carbons in their chain, like 13, 15, 17, 19, and so on. Then there are a couple of other types. One is called very long chain fatty acids, and these are usually more than 24 or 26 carbons in it, and their beta oxidation, half or partial, will happen in peroxisomes, and the other half will happen in mitochondria. And same is the case with the branch chain fatty acids, those that have got some branches in their structure. Their beta oxidations happen in peroxisomes rather than the mitochondria. So the last two basically can be important. Most of the beta oxidations happen in mitochondria, but for very long chain, some part happens in peroxisomes and some part in mitochondria, whereas for the branch chain, the whole process happens in the peroxisomes. So first up is the oxidation of unsaturated fatty acids, and these are the ones that have got a double bond in them, and most of these naturally occurring unsaturated fatty acids have got cis type of double bonds, not the trans. And for the beta oxidation of these unsaturated fatty acids, the main thing that you have to remember is we need two additional enzymes. Sorry. We require all the normal enzymes that we saw in the previous slide for the regular beta oxidation, plus we need two more enzymes. One is isomerase and the other one is reductase. Now the thing to remember is, if it is a mono unsaturated fatty acid, means it has got only one double bond, it will only require isomerase. But if it's a polyunsaturated fatty acids, means more than one double bond, there can be 2, 3, 4, or 5, no matter what number it is, but it has to be more than one double bond. Then we require both the enzymes. Which both enzymes we are talking about? The same isomerase and reductase. So a typical example of the oxidation of a monounsaturated fatty acid has been given, and the example is for the oleic acid, very commonly occurring monounsaturated fatty acid, which has got one double bond present between carbon 9 and carbon 10, also called as oleoyl CoA or oleic acid. Both mean the same thing. And as we saw in the previous slide, the oxidation of monounsaturated fatty acid will require one additional enzyme, that is isomerase. So the full name, enoyl CoA isomerase. And what this enzyme does is it converts the cis isomer into a trans isomer. That is the main function of it. And after the bond has been converted to trans, we will do the regular beta oxidation. So the only problem is for the double bond that is present in this one. So we require an additional enzyme isomerase in it. So here one example of the polyunsaturated fatty acid is also given, and how its oxidation happens. The example is linoleic acid, which has a total of 18 carbons as well, like the oleic acid, but this one has got two double bonds present instead of one. That is why it's called polyunsaturated fatty acid. We can see in the figure one double bond between 9 and 10 and one double bond between carbon 12 and 13. As we saw, for the oxidation of polyunsaturated fatty acid, we need two additional enzymes, isomerase and reductase. So you need to remember the names for polyunsaturated fatty acid. And the combined effect of these two enzymes will be to convert a dienoyl CoA into a trans-enoyl CoA. Simple as that. They will remove a double bond basically. So a trans-dienyl CoA will be converted into a simple trans-enoyl CoA, and after that the regular beta oxidation will happen, like the same reactions, oxidation, hydration, oxidation, and the theolytic cleavage will continue with the regular enzymes of beta oxidation. Next up is oxidation of odd number fatty acids. And odd number fatty acid means that the number of carbons in these fatty acids will be an odd number, like 13, 15, 17, 19, 21, or whatever. One thing to remember is that most of the dietary fatty acids, means the fatty acids that we eat in our diet, are usually even numbered. But many plants and some marine organisms do have these odd number fatty acids. So once they are there, what will basically happen to them? And the main concept to remember is that the process is exactly the same like any even number fatty acid until we reach the end point. So when we started off with the palmitic acid, we saw in every round we take out two carbons in the form of acetyl CoA. So from 16 they become 14 to 10, 8, and so on. And when we had the last four carbons, we break it down into two acetyl CoAs. But the problem here is the odd number fatty acids. So if we start off with 15 or 17 or 19, the same process will continue of the regular beta oxidation. We take out two carbons in every round basically. So say for example we start with 15, in one round we get to 13, then 11, then 9, 7, 5, and then 3. So that is the problem here, that we end up at 3 carbons rather than 4. So that 3 carbon molecule, we cannot directly break it into two acetyl CoAs because it requires a 4 carbon molecule. So the thing to remember basically is that at the end of these odd numbered fatty acid, we get a 3 carbon molecule which is known as a propionyl CoA. So this is the one formed from the beta oxidation of odd number fatty acid, and here it is in the diagram as well, propionyl CoA. And this propionyl CoA will undergo 3 or 4 reactions. So one carboxylation, then epimerase will act on it, and the last one mutase to convert this propionyl CoA into succinyl CoA. So this is what you have to remember. The final product will be a succinyl CoA of this odd chain beta oxidation. First we get propionyl CoA, then it undergoes 3 or 4 reactions to give us succinyl CoA, which can enter the citric acid cycle and give us more energy from it. So the last one for this breakdown is the fatty acid oxidation in peroxisomes. So peroxisomes are a cellular organelles just like mitochondria or nucleus that are present in the cell, and their main function is the fatty acid oxidation of some particular type of fatty acid. So two main types happen in this peroxisomes. One is very long chain fatty acid that have usually more than 26 carbons in them, and then there is the alpha oxidation which is used for branch chain fatty acids, means they have got a branch in it. And the example of these branch chain fatty acids are very important. One is phytanic acid and the other one pristanic acid. So these two are branch chain fatty acids, means they have got a branch in them, and they are usually less commonly found in the dairy products, so they are not very common in nature or in the food, and their catabolism or their beta oxidation requires several additional enzymes. So many other enzymes will be required for their breakdown, and these enzymes are only found in peroxisomes. That is why their beta oxidation mainly happens in peroxisomes. But the peroxisomes also has a problem, that they cannot complete this oxidation because they don't have the enzymes to complete it. They will do the most part basically that is long chain or the branch chain fatty acids will be catabolized to shorter chain fatty acids, which will be around 6 carbons, that is hexonyl CoA. So from 26 or 28 carbons, if it is a long chain fatty acid, these in these peroxisomes they will break them down to 6 carbon chain, which is known as hexonyl CoA, and then it will be exported to the mitochondria, and in the mitochondria the complete oxidation will happen.